Okay. So yeah, thanks again for the introduction. So actually, I'm very happy to be here. Also, I'm kind of a substitute for one person named Steve Piper. Steve Piper is a longtime friend and mentor of mine, and he was meant to give this talk, but he couldn't do it. He had some other appointment. But he's the chief architect of 3D Slicer, which is a very popular open source medical imaging platform for end users. And he's also very involved in the VIZBI community. So he gave a talk 2010 in Heidelberg, I think it was the first VIZBI about physiology and function. And that's part of my talk. I will give a refresher on his stuff. And then there will be part two. So that's me. I'm a research software developer at Boston Children's Hospital. And I'm contributing to Steve's software in my free time right now. And I really love WebGL and JavaScript and web-based programming. So I'm part of the FNN-DSC group, and we do some web-based stuff. So part two will be some live demos of the things we do. And yeah, WebGL, what is it? WebGL is interactive 3D rendering in the browser using your graphics cards. So again, here I'm in the Google Chrome, and I have this WebGL rendering, which is fully interactive here. It's a volume rendering of a 10 months old child. And I can zoom in, I can zoom out. I can use those controls here to go to the slice mode, rotate them. I can go through the slices like this. It runs fast. It's based on OpenGL ES, and yeah, you can threshold on the fly on the shaders. So when we talk about medical imaging, everything starts with the scanner, right? Here you see like the what we call the brain camera for the kids. It's an MR scanner at Boston Children's Hospital. It's we like to make the children very comfortable when they get scanned. We scan all different age ranges, and you operate the scanner from outside from a scanner console, because the room itself has to be magnet proof. And yeah, so the typical data which comes out of a scanner, MR scanner in this case, is like this volume, and there are the slices. You can go through the slices. You can zoom in, and it's typically 1 millimeter resolution and 256 by 256 by 170 voxels in dimensions, and 20 megabyte in size when it comes to neuroimaging. So I'm more focusing on structural MR and diffusion MR, but you can combine different scans, like CT scans, PET scans, and so on, to multimodality imaging, and that's a common approach. For example, to overlay functional information on a structural surface. Here's some MEG data showing responses of audio evoked potentials on the surface. So the MR scanning is very safe for the patient, so safe that we can even scan pregnant women to diagnose the fetal brains. And yeah, when we do that, there's one problem. The baby moves, right? So we get the images like that, which is kind of good in the scan direction, but in the other directions, it looks like that. So it's like unusable. So what we do is some post-processing called motion correction. First, we scan from three angles, and those three images get combined into one using this motion correction approach from Oxford University, and then you get images like that. On the left side here, it's a 28 weeks fetus scanned by us, and yeah, you see the images is all right now for diagnostics, and on the right side, I have the Atlas data from Imperial College, which was built using 50 fetuses off the age of 28 weeks. And yeah, so our physicians use web-based software to compare the scans to this Atlas data. It's easy accessible in the reading room of the MR. And here you can see, I can adjust the window level a little bit. You can see the ventricles in the healthy subject are very thin, but this poor guy has very large ventricles, so it's a pathologic scan, and yeah, that's okay. So when you have those 2D visualizations, which actually the doctors prefer to look at instead of 3D visualizations, you can do a couple of things to enhance the experience. For example, you can adjust the contrast by using the window level function, which you can see here that on the histogram, the gray value scalar just gets compressed, and you have like lower or better contrast. You have corner annotations usually in the software we use, which shows you the current slice, the coordinates, and so on. You can use a color mapping to get a better contrast using rainbow color table, for example. And you can also use the lightbox view. The lightbox view is more the traditional way to look at images, like on film. You know, you have like the same subject, slice by slice next to each other, and you can see changes. So if you adjust one slice, all the other slices jump as well. And a common approach for 2D visualization is labeling the image, and we heard it before on the mice. It's very tedious progress. In our case, also on the images, the doctors or research assistants have to segment every slice. You know, so it can take a week or 2 weeks to segment the whole brain into like different structures and group the voxels based on anatomy or function. And we also have some algorithms to do that automatically, but they are never perfect. You know, for example, there's the free surfer segmentation, which runs 24 hours, and it only works for children larger than 4 years, older than 4 years. So yeah, when you have this 2D visualization, you can make a 3D one out of it. You know, here you see again in WebGL rendered the 3D version of this this segmentation, and that's one approach to get 3D imaging. We also use volume rendering a lot, and there are different methods, but all are based on the ray casting, which shoots like a ray through the images and like looks at the voxels on this ray, and you can sum them all up to have like an X-ray flat image. You can show the one with the highest intensity, which is called a maximum intensity projection. But my favorite is the shaded surface display. And the shaded surface display gets really nice images. You know, this one is a volume rendering, but I wish the resolution would be a little bit better. But it looks real. You know, it looks like somebody just took the skin off. And like I'm very impressed by this research. It's called exposure render photorealistic volume rendering using CUDA from colleagues in Netherlands. And yeah, that's very amazing work. So here are the surfaces. So if you have those label maps, the segmentations, you can use a triangulation algorithm like matching cubes to generate the 3D surface. And then you gain the material properties like opacity, lighting, and can adjust overlays on top of them. Like here we see one with a free surfer mesh with of the left brain hemisphere with curvature overlays. And here's another one. It's a brain aneurysm. And they segmented it and calculated the blood flow. You know, so the brain aneurysm means this guy here, and it can burst if the pressure is too strong. So what they do is they clamp it off, or like do something to avoid the rupture. And here you see the clipping plane where they would clamp it off. So it's like used for pre-surgery. Yeah. So. Another thing is fiber tracking based on diffusion MRI, which shows the white matter pathways in the brain. It's kind of like seeing how the information is distributed in the brain, like information travels, connectivity it's called. And this image shows a tumor with surrounding fiber tracks. And of course, for surgical planning, you need to know which connectivity, which regions are affected, and try to make it as minimal invasive as possible. In 2D, this looks like that you have like the outline of the tumor, and you see the fibers in 2D as well. So before I go into the live demonstrations, I would, in the software, I would first also like to mention this one, registration. So if you have multiple scans of one subject, you can align them properly by registering them. Or if you want to look at like a tumor over time, you do the disease tracking. And there are like mechanisms in the software 3D slicer, for example, which show then the changes. And this tumor here, this was growing, I think 100 milliliters volume grew. And yeah, we heard it before. It's also at the mice. So you can also do group-based analysis by registering many patients to one space or to an atlas to have automatic segmentation approaches. So when we, well, the tools we use is, there are a couple of tools available. This one is 3D Slicer. This one is Freeview, both from Boston. This one is Osiris from Switzerland. It's only available for Mac. This one is TrackViz, and they all do different things. So they're all free available, but TrackViz is not open source. So we are, in our field, we are big fans of open source software and open source algorithms. And here's a list of file formats, which are quite common. So the DICOM file format is the one which comes directly from the scanner. So that's the base. And then researchers convert them to NIfTY, gzip, NIfTY, NERD format, or MGH format to have only one file instead of like a file for each slice. And those are the photo volumes. And for the surface meshes, we use VTK from the visualization toolkit, STL, OBG format. We use this list is the free surface meshes, which is the special software which runs 24 hours, and they spit out all kinds of surfaces. And for the fiber tracks, we either use the same as surfaces, STL and VTK, or this TrackViz TRK format. And if we go back and look at the software, 3D Slicer reads all of them but TrackViz format, the track file. Freeview reads only a subset. Osiris only a subset as well. And TrackViz is the only one who reads the TrackViz format. So if you want to see, for example, a surface mesh and DICOM data enter the tracks, it's hard to find the software to do so. So now I'm showing something live, which we developed at the Children's Hospital. It's called Slice Drop. It's a website. So if we go away from this full screen mode and go to slicedrop.com, it says like instantly view scientific and medical imaging data in 3D, and drop files here or select files. And you see here is other formats which are supported. So I have some data here on my computer, and when I drag and drop it over, it gets loaded in JavaScript, and you can view the stuff here rendered with WebGL. You can do some fiber length thresholding, and this when I dropped it, the data doesn't leave the computer. It just gets parsed and rendered locally on my client. And the slicer works with DICOM data, which comes directly from the scanner. So I can select here some files, drag them over. And here you see the volume got loaded, and you can reorient. That's actually, this orientation should be set automatically, so we're working on that, but that's the tricky one with all the different file formats. So yeah, once you have that, you can adjust, of course, the window level to make the contrast better. You can zoom in. You can switch to 3D volume rendering and adjust the threshold. And here you go. You can look inside the brain. Oh, what's going on? Okay, sorry for that. Was it for the track files? Did you see the track files? No, it's not the power. Yes, that's power. I can go to this. Yeah, so I have to do the window level very strong, so you nearly don't see any contrast anymore. But yeah, you see it better in 3D now. Sorry for that. I don't know why. On my screen it looks fine. So so okay. So yeah, this works on the, and we can also change the color maybe here. Oops. Yeah, so yeah, you get an idea. You can pull your data over to slicedrop.com and view it. And let me try one more thing. the free surfer mesh. If I pull over this mesh with the curvature file, the overlay. Yeah, it's very dark. I don't know. So sorry, I didn't notice that before, but it should not be like that. Okay, well, that was slicedrop.com. So let's see. I also have this one. Oh, let me skip this one for now. Yeah, I want to show this one. It's from UC Berkeley. I'm very happy with the visualization. Yeah, it looks better. So so here you can adjust so what they did was scanned patients with fMRI while showing them videos, annotated videos, and to record which object they see, which region of the brain gets activated. So here are the you look at the brain like that, and I like that you can deflate and inflate it like that, and you can see the semantic information and see which regions of the brain got stimulated by, for example, this one was like the word depository, or like you can choose anything. Aquatic bird. They showed an aquatic bird, and they see which brain regions got activated. And I think that's a very nice example of the WebGL combined with some functional information. And they did a good job. You can change different color tables. Yes. And yes. So another one is this one. It's very dark.
Try full screen.
Yeah, projection mode is better.
Oh, okay. I see. Strange. So yeah, here that's a SPL brain atlas. It's a segmented adult brain with 160 different structures, and you can switch between different views and hover over different structures to see the name. Right here's the corpus callosum. You can go down here to the cerebral cortex and see the different structures. My favorite is the limbic system. You see like the orientation plane here, and yes, rotate. So all kinds of stuff. Hide the orientation layer. And actually, if this works better now, let me just go back to the slice drop. 1 second. So if I, yeah, that's so strange. The full screen changes it. But here you see like there are the overlays. And when you are in SliceDrop, you have those panels on the left where you can like interact live with the data, change colors and all this kind of stuff. So SliceDrop is like a quick viewer for your medical imaging data, because those software packages I mentioned, they are very big. You have to download them, install them, and here you can just go to a website to view the stuff. And let's see. So yeah, I want to show, similar to SliceDrop, we have something called StackDrop. And this is for microscopy images. So it's stackdrop.org. You can select the file. And as I mentioned before, the MR scans are usually like 20 megabyte in size. Here I have like a Z-stack with 1 gigabyte in size. And when you load this Z-stack in the browser as one file, then there are problems. The browser will crash, probably. So we have like some caching. See that? Let me try again. We have some caching to load only 20 slices at once, and you can adjust the cache here. And on my working machine, I can go load to full volume, but here on my MacBook Air, I leave it to 20. And you can see, you can go through all the slices, and it loads now from your file on the fly. So the whole volume never gets loaded into the memory, but yeah, this one. It's a multi-page TIFF format, and I can activate volume rendering, activate adjust the opacity, maybe play with the thresholds, and then of course you can zoom in and rotate this kind of stuff. Yeah, it's very dark again. Yeah. So that's stackdrop.com. And as I said, we like open source software. Like so, each of the things I showed has a source code button in the right, so you can view the source code on GitHub. Yes. So when it comes to the conventional software like 3D Slicer, we have a module 3D Slicer WebGL export. So yeah, you set up your visualization in 3D Slicer and click one button and get the same view as a web page, which you then can share with your collaborators. And it takes into the camera information and everything. And so I don't know if you guys like coding, but most of the examples I showed with the WebGL is based on a WebGL library we developed. It's called XDK, and it's very easy to use. So those are 6 lines of code, and they would show a full-page rendering of a VTK file. Just 6 lines of JavaScript, and you could add another one. You could load a volume, for example, new X volume, v.file, and point it to like a Nifty file. Oops. And then add it as well. And then it will show up rendered as well. The camera, everything gets set up for you, and there are some options. So that's an open source toolkit for doing the WebGL imaging, and it's called XDK, goxdk.com. So when I think about the future, I expect much more virtual reality things to appear and augmented reality. And it's called a virtual holographic display from ZSpace, zspace.com. And in fact, I've seen that last week at Steve Piper's office, and he has a 3D Slicer already integrated, so you can put on like a passive adjustment to your glasses, and then you can see the anatomy in 3D, and you want to touch it. It's like flying in the air in front of you. There's head tracking, so if you move your head, it moves the scene here, and it was very nice and fast. So another one I think is nice is the Oculus Rift VR headset. It was in the press recently because it should be the most responsive headset ever, and will come out soon for a couple of $100s. And I think these kind of devices will change the human-computer interaction. Or like the Leap motion controller, right? I know that Steve Piper already is working on setting this up to control like fiber tracking by your hand. And I think that's a very exciting age right now. And of course, there's also WebCL, which is similar to WebGL, but it's for processing. So soon, instead of just visualization, you can expect to see like image processing in the browser running on your GPU very fast. And yeah, of course, there will be higher resolution, larger data. And yeah, that's it. So those slides are JavaScript again, available on my webpage. And thank you very much.